This video is about how to approach SLOs, or Student Learning Outcomes, also known as Student Learning Objectives. So SLOs are like a roadmap, and the purpose of them is to help focus and to organize content. These are really jam-packed information, and they are going to be very action-oriented, including notice the use of a particular verb, and these are going to take some work to unpack all the different parts to it. They're very carefully constructed to be short, but they do include a lot of information. SLOs, therefore, are not shortcuts, and it can take some practice to understand what to include in them. Your experience in working with them may be something like down here, where at first it's going to be a question like, oh gosh, what's it talking about? Then you need to think about it a little bit. Hopefully you get some inspiration about what the answer is, and then you can come to the conclusion. Realistically, though, it's not linear in this respect. A lot of times you will come to this point here, then you should confer with other people, like your professor or your peers or a study group, or just give yourself some time and then look at it again later, maybe in an hour or the next day, and then come back to it and think about it some more, and then keep working on it. SLOs are also part of the growth mindset, so try to keep this idea that you're working your brain out as you are working through these, and you are being asked to do things that you don't know the answer to. The idea of this is that it helps you to create new neural connections in your brain, and so this increases and enhances your learning, not only in the short term, but also in the long term, basically so you can learn it better. So here's an example of an SLO. This is SLO number 9 from Unit 2 of Biology 201, and it states here to explain the laws of heredity and how they relate to genetic inheritance. It also has this blue dot here. So I'm going to talk some more specifics about this after I talk about a general way to approach an SLO. Here's some general tips to approaching how to break down the SLOs. Notice that this is an active process. At the beginning part of breaking down the SLO, you should read it over and then identify the verb for which type of information to include. Some verbs include list, in which case you really just write out the terms. Other ones might say to explain, which means this is looking for a longer, more sentence-driven process. It probably will take several sentences to answer it completely. Or it might be to draw, which means you should be able to draw it from memory. Or it might say to label, which means that you would be given the drawing and then you would write the labels on it. You should also notice that a great way to study is to try to use the SLO and use different verbs so you can continue to practice the information. Also note the color-coded dots that we use in Biology 201. This is to use to gauge the depth of information that we expect you to know for that particular topic. The basic scheme is there are either blue or purple dots, and those are more introductory information, and so more about terminology and basically memorizing the information. Then when you see green dots, either light or dark green, that is higher level information to know, which includes analyzing the information or applying it. And so that means that you need to read into it a little more, and it's more of a puzzle to figure out. Finally, for getting started with the SLOs, you should identify the definitions, maybe underline those or circle them, and then write out the definitions for each terminology term that is used in the question. This includes even kind of simple things like the process. So process is also a clue for this sort of thing that we're looking for multiple steps and how they fit together. Okay, then once you have identified those things, then you should go on to the next part, which would be to identify the corresponding information in your resources. You have several different resources to pull from, including your lecture notes, the reading guide, the text, and handouts. And so basically you want to have that SLO in front of you and then pull out your resources and try to find it within one or more of those resources. And a good idea would be to look through all of them to see what you have available to you. One warning is that the terminology does not always match perfectly, and so sometimes you need to kind of read between the lines to be able to match up the terminology that's, say, in the textbook versus in your resources. And this is a really good time to ask your professor or your peers what they're finding for that. It's a great time to check in. Next step is to incorporate any additional associated terminology, other words that are used within the reading guide or textbook in your responses. This is a way to increase the depth of your information, plus expand it to other content. You should consider using drawings with labels as well as written out information, for example, as stepwise phrases, step 1, step 2, step 3, with those corresponding numbers on your drawing as different ways to answer the SLO. This depends on what you really study well from. Everyone should benefit from doing both of these things, although some people may have a preference for one or the other. Finally, you should note which references that you used off to the side. For example, Reading Guide 16, Handout 16, Text Page 78, Lecture Notes, and whatever date. This way you could go back to them if you needed to. And overall, you probably need to go into more depth than you think you do for a SLO. So be sure that you really are complete. And this is where it's really useful to come back to this information later so you can start to bring in more details. It's also a good time to check in with your peers so that we can kind of cover all the bases and get a complete view of the process. Okay, so let's break down this one. Basically, let's start off with looking for the verb. So as we look at this, so explain is our verb here. And then let's start to underline some of our vocabulary. Laws of heredity and how they relate to genetic inheritance. And probably those are two different terms. Also note the blue dot, so this means that this is a little more of the kind of introductory information that you should be able to find directly in your notes. Explain also is going to refer to several sentences to write up to answer it. All right, so if you pull out your textbook, basically you're going to find that it doesn't use the word laws of heredity. Instead, it uses principles. So this is one of those cases where the terminology is a little bit different. In the lecture notes, I used both of these terms, laws and principles. So if you look for that source, you should be able to tag each of these. And so when we're talking about heredity, we are talking about how information is passed down from one generation to the next. And then let's pull out a couple other things here. So genetic inheritance. So genetic means we're talking about genes. This means that we're talking about DNA and or chromosomes. And again, inheritance plays off this term heredity, talking about how information is passed down from one generation to the next. Also notice this verb over here, relate. So that means that we're trying to connect up these two concepts, the laws of heredity with the genetic inheritance. Okay, so let's talk about what those laws of inheritance or principles of inheritance are. So first one is the principle of segregation. And the second one is the principle of independent assortment. Don't forget that we want to make some notes about where we're finding these things. And so basically a couple points that I will call your attention to are the introduction of chapter 14 from your textbook. This is basically right before section 14.1. And then the principle of segregation, this is in the textbook, section 14.2. Also in the textbook is principle of independent assortment, which is textbook 14.3. Okay, so principle of segregation. This has to do with how one copy of each gene is packaged into a gamete, which would be the egg or sperm, during meiosis, which is the process of making gametes. Okay, so let's add a little bit more to this. So one copy of each gene. So another way to say that is an allele. And kind of a spin-off of this is that each gamete has one of the two copies of information that the parent had. And the big idea is that they are segregating from each other. So we start with two copies, and those two copies are pulled apart. So basically the alleles are pulled apart from each other. And later we're going to find out there's a physical process to this, but for now, the idea is that this is going to result in each gamete only getting one piece of information. Okay, now let's talk about the principle of independent assortment. And this is when each gene is packaged independently of other genes during gamete formation. And so let's add in a little bit more information about that. So each gene, let's say that this is the R gene with respect to other genes, for example, like the Y gene. And so this results in an assortment, basically, of a new combination of these genes. And basically these are independent of each other. So the R gene versus the Y gene each do their own thing. And so picking, say, the dominant allele for the R gene does not mean that we would also get the dominant allele for the Y gene. It's a coin flip for each of those. And since we've put some letters to our principle of independent assortment, let's do the same thing up with the principle of segregation. So for the principle of segregation, this has to do with a particular gene, just one gene, and its alleles. So let's put a little example there. So let's say that we're talking about the R gene here, and what are pulled apart are the big R allele versus the little r allele. And we're just considering one gene at a time for the principle of segregation. On the other hand, we're considering two genes at a time for the independent assortment principle. Okay, so I hope that helped to see how I brought some more information in for each of these. And so notice that I kept going back after I wrote my definition, and then I'd fill in some more information. Sometimes I worked with the terminology here by just writing it out in parentheses. Other times I wrote it off to the side. You want to give yourself a lot of space as you work through these SLOs, obviously, and also give yourself some time to be able to go back and look at them again. And then I want to wrap up to talk about this relate part. So here we spent a lot of time talking about defining the laws of heredity, which were up here, those two laws. So let's talk about the relating part. That's basically what I've written in over here in green. So how does that relate to genetic inheritance? And basically how does it relate to genes and how they're passed on to the next generation? So basically talked about how each gamete has one of the copies of information that each parent had. And so this is basically what we would say in everyday life, that you get half your information from mom or half your information from dad. And so right here, we're just talking about the half from each parent. Eventually, of course, those will combine together, but right now we're really just talking about pulling things apart. So this is very simple core concept that we kind of skip over and take for granted just from what we know in everyday life nowadays. On the other hand, for the second principle, to talk about independent assortment, this is the big idea about relating to genetic inheritance, that there are new combinations of these genes in an individual, and so we could get combinations that were not seen before in the parents. And ultimately, why this is so important to genetic inheritance is that this leads to diversity in terms of combinations of information. And let's say that one of the parents was big R, little y, little y. The other parent was little r, big Y, big Y. So this would be round and green, and the other parent would be wrinkled and yellow. And basically then, when we look at the offspring for this guy, then we're going to end up with big R, little r, big Y, little y. And so now we're going to end up with offspring that have both dominant traits, namely that they'll be round and yellow, which is completely different than something we've seen before, which was round and green versus wrinkled and yellow. And so I know that doesn't sound super exciting, perhaps, but think about this for other traits, and you may realize that this could end up being some really powerful combinations. Sometimes, other times not so great, but the big idea is that we have diversity and variation across a population. So that's a really big concept that we'll hear more about later in the course.